one doesn't start and get lupus bang on Tuesday of this week. It doesn't suddenly explode. A lot of people say, well, I got this bad flu or this bad cold, and then I got lupus. It doesn't work that way. Actually, if we could look, I'm going to make an example of you, I don't mean to, but let's say if we could look at your blood 5 or 10 years before you had lupus, you had those antibodies that Laura was talking about, those anti-nuclear antibodies. Almost certainly, I bet. Okay. And so that the shift from genetic susceptibility, you've got the genes, which is your question, and the first thing that goes wrong is that autoimmunity and autoimmune recognition, as you said, you kind of don't know that it's yourself, so you start attacking yourself, but not seriously, very gently. So they're just little antibodies, and you're perfectly well. No one would know it except that you could find a lab test. So the first thing we would do in a family like you is to check the family members and check their ANA, even though they're totally healthy, because that could be the start of lupus. Now, if you know, could be, and because of the very positive family history that you have, you would take that very seriously, that everything might be conducive. If we just screened 1,000 people and we found those few people that would have just ANAs generally, we wouldn't know. The odds wouldn't favor it, but in your case, within the family, they would. So we might suppose we found, I had a sister, let's say, and she was ANA positive, but that's all. No lupus, perfectly well. Well, there are drugs, one of them hydroxychloroquine that most of you know, that we might well start that person on. I mean, it's a serious decision, and you could argue one way or the other, but we think a lot about it. Of whether to try to prevent. We'd certainly say use high SPF clothing that is going to keep the sun off. Be sure you wear sunscreen, long sleeve shirts that are going to be protective. Don't smoke. That's very important. Okay? So we'd say those things right away, because each of those things are going to reduce the likelihood of this eventuating into lupus. So we have a window of some years to deviate this. How successful? I don't know. But 10 years ago we didn't know that. It really wasn't well appreciated. Now it's known that the
20 years ago it was.
yeah, no, certainly wasn't. There's always been, in part, lupus has been intellectually a very, very challenging disease for doctors. So it's been difficult, and there are some people that like difficult challenges. So I'm old enough to have gotten into the study of lupus. There wasn't that much we could do. There was steroids and maybe Cytoxan, but it was a challenge to diagnose and a challenge. So that's what caught some people. And there was always research done on lupus, but the Lupus Foundation, in particular, the various lupus foundations, have really galvanized this so that the amount of research is greatly expanded. And not only just the amount of research, but the ability to translate from something that might be done for diabetes into something that's done for lupus. And in fact, this genome effort that really uncorked the bottle that let all of this happen shows us that the genes for example, multiple sclerosis, which is an autoimmune disease, some of these genes are shared by lupus. Some genes that cause multiple sclerosis are not found in lupus, but they're found in rheumatoid arthritis. So each of the autoimmune diseases shares certain genes, but doesn't share others. So that knowledge means that many of the studies that are advances in the field of diabetes become advances in the field of lupus, because it's the same gene.
Oh, okay.
Okay, so it really, it's an explosion of knowledge. I can't keep up with it all.
Okay.
You really have to talk nowadays in thousands or tens of thousands or 20s or more thousands of patients. So most of the major studies in lupus now involve multiple centers. So for example, could be at Columbia if we're not so well organized in terms of our patient population here. But in some other centers, they're very well organized, so they may have 100 to 200 lupus patients and all their family members. So the DNA from those people goes to a central repository and everybody exchanges. And because of the Human Genome Project, we know the code. We can figure out, so at position milestone 10,500 or some arbitrary number, we know there should be a certain gene, and that's a gene that confers susceptibility on lupus. So everybody uses the same technology in Sweden, in Germany, in Ireland, in England, in the US, in Canada, multiple places. You all get together so that you end up with a consortium of maybe 30 people or 20 people that have done this, basically everything, because what you have to do is replicate and repeat to make sure that it just isn't a statistical hoax. And so you have validation cohorts, and then you put them together different ways, different ethnic groups, different populations. And this becomes the body of knowledge. And then there are many, many repositories. So I mean, there's just a beautiful paper together that looked at where these genes are expressed in what kind of cell, because to find out how they work, you need to know where their place of employment is. So there are 200 or 300 different kinds of cells in the body. And for example, a lupus cell, lupus genes are mainly working in B cells, the abnormal genes, to a lesser degree in T cells. In rheumatoid arthritis, they're in a special subset of T cells, but not in B cells. So the genes that confer predisposition to a disease, we now, but by virtue of these large data banks, you can put them in a cell type, and you can begin to figure them out. You can use a mouse experiment because some of the genes are the same in the mouse and the human, so you can sort it all out that way.
But
So
the sample size has gotten pretty big.
the sample size has got to be enormous. Enormous. Really, 20s, 30,000 are what you're seeing now. And that's how it happens, through collaboration, through research supported by the foundation and by the government, at least when it was supporting research. So it isn't environment in the sense that I might walk down the street and the flower pot falls off a window ledge and hits me on the head. That's one kind of environment. But it is environmental in terms of infections, as we heard, or smoking, that are going to bring out this genetic tendency. So presumably, I bet if we went into your DNA, you'd have these genes. They'd all be there. They weren't expressed in either parent because maybe the total number of them wasn't quite up to that threshold that gives you lupus, or maybe they were there and you never developed disease. The best example of that are identical twins. So if you look at identical twins from birth, follow them, let's say one develops lupus maybe age 25. Now they're genetically identical, the environments are pretty much the same. Well, the other person isn't necessarily going to develop the same instant. So you follow, and if you follow a few years, you'll find that what we call concordance, the fact that two patients have developed the disease, is maybe 25%. If you follow longer, it gets as high as 60 to 65% if you follow 25 or 30 years. Never 100. Now they're genetically identical. But they're both ANA positive. They have autoimmunity, but only one has gone on to the disease. Our immune system is trying to help us to the same extent as trying to hurt us. So it could be that they haven't quite got to these checkpoints, they haven't got enough genes. I mean, I can only speculate. But I'm sure that if we looked at you, we'd find these.
So I could still blame my parents.
I'm sorry,
Yes, I can still
blame,
blame.
because you have to be very careful with your parents. It's
Wow.
important.